Now, let's see the naming conventions of these amino acids, right? All amino acid side chains except glycine have one or more carbon atoms. These are named sequentially using Greek letters. Branch groups get a prime or a numeric designation. Now for lysine, as you can see, it's a carboxylate and it's an amine, and this is the alpha carbon. All the subsequent carbons have been designated by the Greek letters. Now, since this is alpha, this is beta, gamma, delta, and epsilon, right? And this is the R group that we have seen before. Now, if it's a branched chain, you will have additional designation of 1 and 2. Now, it's time that we get to know these amino acids. Now, I do not have to tell you this, but it is important that you understand and remember the names and structures of these amino acids. It would be much easier if you remember the structure of the R groups so that you can compare and contrast. And that's why we are looking at R groups here. Since most of the amino acid structure is the same, the R group or side chain is what imparts unique chemical functionality to amino acids. The 5 major classes of amino acid R groups. 1. Nonpolar, aliphatic, hydrophobic. There are 7 of them. Aromatic, 3 of them. Polar, uncharged, 5 of them. Positively charged, 3. Negatively charged, 2. If you combine them, 2 plus 3, 5, 5 plus 5, 10, 10 plus 3, 13, plus 7, 20. So all 20 amino acids are accounted for, but they are divided into 1, 2, 3, 4, 5 groups. The easiest one to remember, the positively charged, negatively charged, and the aromatic ones. The polar, uncharged, and non-polar may not be that easy to remember, but there are ways to remember these structures and names, right? And I'll explain it to you as to how you can do that. Let's look at the first group, nonpolar or aliphatic and hydrophobic. So the first one is glycine. The second one is alanine. The difference is that for glycine, it's a hydrogen. In alanine, it's a methyl group. Then comes proline. Proline is an exception here. If you were to look at it, proline has this cyclic ring, right? Now the cyclic ring extends into this amino group. So the NH2 here is a secondary amine as compared to all the other 19 amino acids, which are primary amines, right? That's the exception here. Fourth one is valine, right? It's an isopropyl group. Then leucine, another methyl added to the isopropyl, isoleucine. Now the difference between these two are isomers. Now the isomerism is because the methyl is attached here for isoleucine, whereas for leucine it is attached here. And then comes methionine. Methionine again is different from others because it has a sulfur. So these are the different amino acids, and here we will be introduced to the branched amino acids, right? Valine and isoleucine, right? Valine is branched, isoleucine is branched, as well as leucine. Leucine is gamma branched, whereas valine and isoleucine are beta branched. That's the difference. Now proline, like I said, has a secondary nitrogen, secondary amine group, and is sometimes known as the amino acid. Now these amino acids, aliphatic, nonpolar, are found in the interiors of folded proteins. Why? Now that's a question that you need to ask yourself and answer it. Right? I will be able to help you out with this when we talk about this later on. Right? That's an interesting question, and you know the answer because we looked at this in chapter 2. Right? Think about it. The second group is aromatic. Aromatic groups impart strong absorbance in the UV range. And there are three aromatic amino acids. The first one is called phenylalanine. Now, you know what alanine is. Alanine is with a methyl group. Now, if you replace one of the hydrogen with a phenyl group, it is phenylalanine. It's easy to remember, right? Now, if you add a hydroxy group to phenylalanine, it becomes tyrosine. So these two structures are easier to remember as compared to others, right? Alanine, phenylalanine, and tyrosine, where phenylalanine and tyrosine are aromatic amino acids. Tryptophan. Tryptophan is an outlier because it has an indole group. The R group is an indole substituent, right? So tryptophan is a very it's an outlier, and it's a very important amino acid. We'll talk about this. Tyrosine is polar because in the sense that it has a polar functionality, right? Well, phenylalanine does not have a polar functionality and is completely hydrophobic. Tyrosine can at times poke its head into the polar regions using its hydroxy substituent because this OH can hydrogen bond with water molecules and other polar groups, right? Tryptophan is strongly fluorescent while tyrosine is weakly fluorescent. So tryptophan is the only amino acid in proteins that is fluorescent. Now let's look at the third group, polar uncharged amino acids. There are 5 of them here, right? They have slight preference for the exteriors of folded proteins. Polar groups are often the site of hydrogen bonding interactions. Cysteine is involved in disulfide bonding. Asparagine and glutamine are analogs of aspartate and glutamate. Now, we'll look at as to what aspartate and glutamates are, right? Now, the first amino acid that I want your attention is serine. Now, what is the difference? Now, serine, in this case, has a hydroxy substituent attached to this methylene group, right? Now, if there were no OH and it's just H, it is alanine, right? If you replace that H with an OH, it is called a serine. Threonine is when you have another carbon attached and a methyl, and in this beta carbon there's a hydroxy substituent attached. So this threonine is a branched chain amino acid. The branch is that it is a beta branched amino acid. It's very similar to valine. The only exception is that that methyl group in valine is replaced by a hydroxy, and hence the name threonine. Cysteine is another amino acid where one of the hydrogen in alanine is replaced by a thiol or SH group. So the difference between serine and cysteine is that serine has an OH and cysteine has an SH, right? And cysteines are involved in disulfide bonding. And what is disulfide bonding? And we'll talk about this later on. 2 other amino acids, asparagine and glutamine. Asparagine has an amide substituent in its beta carbon, whereas glutamine has an amide substituent in its gamma carbon, right? This is alpha, beta, and this is alpha, beta, gamma. So its amide is CONH2, and hence the name asparagine and glutamine. What about positively charged amino acids? Often found in the exteriors of folded proteins. Again, think about it. Why? 3 different positively charged amino acids. One is lysine, another one is arginine, and the third one is called histidine. What is the difference? Lysine has got a long alkyl chain. At the end of the alkyl chain is a primary amine that is positively charged, right? Arginine, on the other hand, has got this 3 alkyl chain, whereas lysine has 4 alkyl chain, 1, 2, 3, 4, and arginine has 1, 2, 3 attached, and to the third carbon, or the alpha, beta, gamma, delta. Delta carbon is a guanidinium group, and hence it's called as arginine. It's positively charged. Histidine, on the other hand, is where you have an imidazole substituent, an imidazole functional group attached to the beta carbon. Now, histidine has a pKa that is close to biological pH. It's about 6. Now, we'll talk about as to what the pKa is and what are the consequences of the pKa of side chains or the R groups of amino acids are and stuff like that a little later. Now, let's look at negatively charged amino acids. There are two of them, aspartate and glutamate. Again, found on the exterior of folded proteins, sometimes on the interior as well. Both have carboxylates. So aspartate has a carboxylate attached to the beta carbon, glutamate on the gamma carbon, right? The pKa's are very low, but very close. This has a pKa of 3.9 and glutamate has a pKa of 4.1. And mind you, all of these are pKa's of the side chains. Now, aspartate and glutamate, the names are pretty similar to asparagine and glutamine, right? And hence, when we looked at the previous slide, asparagine and glutamine were called as the amide derivatives of aspartate and glutamate. And asparagine and glutamine are uncharged, whereas aspartate and glutamate are negatively charged. One more property of aromatic amino acids is that they have UV absorbance, right? It's a common way, UV absorbance is a common way to determine the concentrations of protein in solution. And you learn about this when you do labs, right? We typically measure the absorbance at 280 nanometers. That's where the maximal absorbance of tryptophan is. Right? Remember, tryptophan has this indole moiety. There's a tyrosine and phenylalanine. And if you look at the absorbance spectra, right, the absorbance of tryptophan is really high at 280 nanometers. Tyrosine, not so much, and phenylalanine is very less. Again, reiterating the fact that aromatic amino acids absorb light in the UV regions, and proteins typically have UV absorbance maxima around 280 nanometers. Tryptophan and tyrosines are strong chromophores. Remember this, right? Now, concentrations can be determined by UV visible spectrophotometry using the Beer-Lambert's law. That is, A is equal to epsilon LC, where A is the absorbance, E is the molar extinction coefficient, L is the path length of the cuvette, and C is the concentration of the sample. And in this case, you will be measuring the concentration of your protein using the UV absorbance by incorporating the molar extinction coefficient and a specific path length.